Welcome to the Neural Implant Podcast, where we talk with the people behind the current events and breakthroughs in brain implants in an understandable way, helping bring together various fields involved in neuroprosthetics. Here is your host, Ladan.
Hello, everyone, and welcome to the Neural Implant Podcast. Today's guest is Stavros Zanos. And the keen-eared among you might think, wait a second, we had a Zanos on the show already. We had Theodoros Zanos on the show almost 2, maybe even 3 years ago. What is going on? And you'd be right. And I'd be impressed with how well you followed the guests on the show. And they are, of course, brothers, and they both work at the Feinstein Institute in New York, the same place that Kevin Tracy, a previous guest, put together, basically Institute of Bioelectronic Medicine. And in this show, we're talking about a new paper that's being put together with anodal blocking of the vagus nerve. And this basically is able to determine or select which side, afferent or efferent, efferent going away, emitting, afferent going towards the brain, accepting. And they're able to selectively block either one of these. So that's really cool, very cool for the field of bioelectronic medicine, a little bit more selectivity. So yeah, we talk about this and some of their work at the Feinstein Institute. Enjoy. Stavros Zanos, pleasure to have you on the show. You are an assistant professor at Feinstein Institute. I think you officially make a full house, like in poker, right, of Feinstein guests. I think you're the fifth one. Pleasure to have you on the show. We've had your brother on, Theo Zanos, so we're going to talk about this a little bit later. But we're talking to you because you have published recently very interesting results in Nature Scientific about anodal blocking in the vagus nerve. And so obviously you've been working a lot with bioelectronic medicine, which is a huge topic on the show and a very popular topic on the show. But do you want to introduce yourself?
Yeah, so I'm Stavros. I joined Feinstein. I moved to New York from Seattle, where I was a postdoc with Eberhard Fetz at the University of Washington. I also did my PhD with Eb, all that background in primate neurophysiology and brain-computer interfaces. I also came to Feinstein with a background in clinical cardiology and medicine. I'm also a physician. So Feinstein was a choice for me because it is a research institute affiliated with a big health system in New York. And their approach to research is very translational. Even though in my PhD and my postdoc with Eb, I did a lot of basic science. Like I said, I also have that clinical side that I would like to combine with the scientific side. So Feinstein was an obvious choice for me. And I've been having a lot of fun for the past 3 years.
Yeah, it's pretty cool. And what would you say your research focus is and your brother's research focus? You and your brother are working in the same institute. That doesn't happen often. That's really cool.
Yeah, Theo joined Feinstein about a year before I did. And, as you can imagine, he did play a role in me deciding to come. So my focus since I joined has been, well, first I had to organize the animal lab here. So we spend a lot of time building the rodent facility, let's say, lab with physiology stations. And then we also build the large animal lab as well. We don't have monkeys here, but we do have pigs. And it turns out that pig is a pretty good model for bioelectronic medicine. I didn't know it back then. I found out later. But that was a fortunate event. So the focus has been bioelectronic medicine, and in particular, the animal testing, engineering of implants, design of stimulation waveforms, understanding the physiological effects of these bioelectronic therapies in these animal models, developing some chronic implants for testing therapies in animal models. So I would say, in a nutshell, it has been an approach that combines neurophysiology with neural engineering and translational research. And all of that revolving around the idea of stimulating nerves or neural tissue in general to treat diseases. And those diseases could be diseases of the nervous system, or it could be diseases in which the nervous system is implicated in. So not only brain or spinal cord or even peripheral nerve diseases, but diseases of the spleen, autoimmune diseases of the liver, like metabolic diseases like diabetes and obesity, disease of the cardiovascular system or the cardiopulmonary system.
Yeah, very cool. Definitely a lot of vagus nerve stimulation. I mean, that kind of affects everything. It doesn't necessarily need to be nervous system related. Do you want to talk about this finding with the anodal block? And first of all, what is anodal block? What is this newest publication from you guys?
It was part of a bigger effort to build the tools, I would say initially, for some rational vagus nerve stimulation therapies. When I say rational, I mean designing therapies that target specific elements of the nervous system, in particular the vagus nerve, with the mindset that we're trying to maximize efficacy and minimize side effects. And that's important for the vagus in particular because it has a lot of different fiber types. Not all of them should be stimulated for all diseases. So, behind this idea of selectivity, we had to come up with ways to deliver smarter therapies, better therapies. And a lot of the principles behind that way of thinking are not new. They've been around for decades in peripheral nerve stimulation. It's just that some of them have not been applied as rigorous in the bioelectronic medicine and vagus nerve stimulation in particular. So, one of those ways to be more selective with VNS was this, I would say, biasing the activation of afferent or sensory versus efferent or motor fibers. If you are trying to treat a disease of the brain, you probably don't want to stimulate the peripheral organs and vice versa. If you are after bioelectronic therapy of heart failure, for example, you don't want to stimulate the afferent system too much unless you have to. So this anodal block is really a way to bias the activation of fibers towards the efferent or the afferent direction. And we can get into details about the biophysics of it. You rely on a hyperpolarized patch of membrane, and that's the anode, that blocks the conduction of action potentials. So if you have that patch of membrane that hyperpolarized on the rostral side of the electrode, then you're blocking the action potentials that travel towards the brain. If you place it, you have it on the caudal side of the electrode, you're blocking action potentials that travel towards the peripheral organs. So that's the idea behind this directional biasing. Like I said, it wasn't a new idea. It has been shown to work in peripheral nerves, somatic nerves mostly. In the vagus, it was not clear whether it was working. Some people claim it worked, some others claim it didn't. So while we were developing these methodologies to develop these directional or fiber selective waveforms, and we became interested that does this really work or not? And that formed the basis for the paper. Just to give you a very brief overview of what we found, a nodal block as a mechanism does work as intended in the context of vagus nerve stimulation. So we showed that it's a useful technique to bias directional activation towards the afferent of the efferent side. And by the way, clinical therapies of VNS, like as it's used for epilepsy and as it has been tested for heart failure, did make use of this principle. It's nothing novel. This mechanism does work as it was intended to work.
Okay. Yeah, because if I remember correctly, the vagus nerve is actually mostly basically motor neurons sending out, and only I think it's something like 10% afferent. So if you're able to, change which one, I mean, you're able to eliminate like 90% and continue function with 90%, right?
Yes, exactly. The percentage of afferent and different fibers in the vagus is a matter of debate. I think it's probably different in different animals. Things change all the time, but my impression is that most of them are afferent and smaller size fibers. But the efferent fibers are usually the ones that get activated first because at least some of them are very large. So the way that the vagus gets stimulated, and all nerves really, is at the lowest intensity of stimuli, you have the larger fibers get engaged first. And then as you increase the intensity, you engage smaller and smaller fibers. Well, some of the largest fibers are these motor fibers that innervate the laryngeal muscles. So you generally don't want to activate those, but that is not easy because they are the first one to be activated, right? So that's where all these ideas about selectivity and fiber specificity and stuff like that come in.
Okay. Yeah, it's a bit confusing, the efferent, efferent. I don't know. I always have to be like, okay, wait, which one is it again? How are you guys going to use this technology? What does this mean for the field to be able to have this kind of selective blocking?
I would say it's a first tool in hopefully in a range of tools that we will have or that we have to create better therapies that are based on vagus nerve stimulation. I mentioned the example of the fibers that innervate the muscles of the larynx and how you don't want to activate those. But there are other fibers that innervate the heart, for example, and those are pretty small. So you need to get to those diseases like arrhythmias or heart failure. At the same time, you don't want to cause bronchospasm, for example, because there are other fibers that innervate the lungs, in which, when they're activated, and those are also efferent, right? They cause the bronchi to be constricted, so that can give people an asthma attack. So it's just one of the many tools that we are trying to develop to get better selectivity with regards to which fibers we're activating, and thereby increase the therapeutic index, as we call it. So the range of dose intensities that give us a good effect versus those that give us a bad effect. We want those two to be as different as possible. So that's what we've been working on. And it's not just us, it's a lot of labs around the world and labs with a big history in the field that have been developing different ways of maximizing this selective neurostimulation.
What kind of improvements would you expect, or did you guys measure?
Well, in this particular paper, a somewhat novel aspect of it was that we didn't only rely on recording evoked neural activity from the stimulation, which is something that the field has been doing for many, many years. And it is the golden standard for assessing the effects of neurostimulation. We went one step further and looked at what happens at the end organs, right? So if we do activate the efferent fibers, what kind of physiological changes do we expect to see? And the same for afferent fibers. So we did that by a very crude but very useful tool or technique, which is called the vagotomy. So you can cut the nerve, and if you cut it above the cuff that you're stimulating, then you obviously interrupt the afferent communication to the brain. If you cut it below the cuff, the stimulating cuff, you interrupt the efferent communication to the rest of the body, right? So we did that. We stimulated the nerve before and after the vagotomy, and based on what effects disappeared from the vagotomy after the vagotomy, we could tell that those effects were either afferent or efferent. So we found that changes in heart rate that happen with VNS disappear when you do an efferent vagotomy, like a caudal vagotomy, and changes in the way the animal breathes disappeared after we did the rostral vagotomy. So changes in breathing and changes in heart rate are good indices, physiological indices of afferent and efferent fiber activation. So we use those to compare the two different polarities that go into implementing this phenomenon of anodal block. And that's where we found differences. So in one configuration, we got a stronger efferent effect, meaning stronger changes in heart rate, weaker changes in breathing. And in the other configuration, we observed the opposite. So stronger changes in breathing and weaker changes in heart rate.
What are some difficulties with this? I mean, it sounds like you kind of have to tease out some result. What are some challenges with this work?
Well, I would say the major challenge is the complexity of the effects. It's not just different changes in heart rate and breathing. Right? There are a lot of things changing with VNS, and some of them confound each other. So you need to be very careful about dissecting the different effects. I would say another difficult part that we had to do, though, was while registering all these physiological responses, at the same time, we had to measure evoked potentials in the nerves. And that's not easy. The people who did the experiments were complaining that in those times that got amazing physiological effects, they couldn't record evoked potentials, and vice versa. In the cases where they got amazing neural responses, they couldn't see physiological effects. That was in the beginning, and then as we all became better and our tools became better, We reached a point where we got almost 100% in both. But that was a challenging part. The confounding effect of anesthesia was, I think, a big deal. We're looking at slightly different physiology when the animals are anesthetized than when they are conscious. And we always have to remember that when we try to extrapolate findings from animals to humans, because humans receive VNS even when they're not unconscious. How do our findings translate to the human condition? We always have to be reminiscent of the conditions under which the data were collected. And of course, the differences between species. So we do a lot of our experiments in mice, rats, and pigs more lately. And those have a lot of advantages and limitations. Each model has its own advantages and limitations, but you have to be very careful about what is translatable and what is not translatable. And I think one of the reasons why you should be careful is for saving time, really. You can spend years testing therapies in mice, and in the end, very few of those translate into therapies that are effective in humans. I think there are ways, at least the way I see it in bioelectronic medicine and neuromodulation in general, to still use animals as screening tools, but be a little more careful and a little smarter about what aspects of these therapies are likely to be translated and which ones are not.
So these tests had to be done with anesthetized animals. I mean, would that be different? You mentioned kind of the difference between humans, also, being awake. Would you be able to do these experiments with awake animals or not?
We have done it. We do get very similar results. And actually, there is a postdoc in the lab that is writing a paper on that. That required developing chronic implants because you need to repeat the same experiment at least twice. Once with the animal anesthetized and then with the animal awake. So we have developed these chronic implants in rats and now recently, less than a year ago, in mice. So we can test all of these, we can ask all of these questions in our animals. To answer your question, yes, it does make a difference. I would say overall, it's a little easier to evoke certain physiological effects when the animal is awake. Maybe not all, but some obvious physiological effects definitely it's a little easier to evoke in conscious animals than it is to evoke in anesthetized.
Okay. And then you were talking about chronic implants. I mean, would this change over a longer period of time? I mean, you have some human patients, I think, too, right, at Feinstein, and they obviously want to have, longer implants, more than, I don't know, a week or something like that, right?
Yeah, no, these vagus implants that are used for the treatment of epilepsy, they last for years. So that has been another major focus of the lab to develop these chronic implants in animals and then understand how to make them last longer or how the interface changes over time. Like I mentioned before, there is a current effort in the lab that resulted in a preprint, but soon to be submitted as a paper that characterized the long-term performance of a cervical vagus cuff in a mouse. Mice are very small and their vagus nerves are even smaller. It was challenging. We did have to collaborate with two other labs on this. One is in University of Colorado, Christine Welles' lab, and the other one is at New York University, Robert Franke's lab. They both were developing, similar techniques to interface with the vagus chronically. So we collaborated with them, and like you said, the performance of these implants, we found that changes over time. It generally becomes harder and harder to engage the same, let's say, number of fibers as time progresses. But there is a way to account for that. So you just have to define standards for VNS dose that you just have to track as the interface evolves.
And then do you want to describe how the implant works, or the blocking works in a little bit more detail? You mentioned it a little bit earlier.
Yeah, so basically we use these bipolar electrodes. One of them serves as the cathode. So you depolarize the membrane at that point, and that's where the action potentials are initiated. And then they start traveling in both ways. That's how it works with axons, right? So one way is towards the brain, the other one is towards the periphery. As they travel, they either encounter, let's say, membrane of resting potential, in which case they just keep going. Or in some cases, they might encounter membrane at the hyperpolarized potential. Now, we make sure we place our anode at the right distance from the cathode, so it creates this hyperpolarizing patch when the action potentials from the cathode reach that patch. So when they do reach that patch, they encounter the hyperpolarized potential, so a lot of them get blocked. Where there is no hyperpolarized patch, they just go right through. So we can increase the intensity of stimulation and get more and more potentials going in one direction, and then blocking some of the potentials. It's never 100%, but some of the potentials in the other direction. And this phenomenon has some parameters that it works well with and some others that it doesn't. The width of the depolarizing pulse is very important, and the relationship between that pulse width with the conduction time between the cathode and the anode. So it requires a little bit of tweaking there. Also, the intensity, because, like I said before, the intensity determines which fibers, the size of the fibers that get engaged. So the lower the intensity, the larger the fibers that are engaged, but those are also tend to be faster. So there is a combination of parameters that affect whether this phenomenon of anodal block is successful or not, based on distances and conduction times and the types of fibers that get recruited.
Okay, cool. And then what are some short-term and long-term plans for you guys in this work?
Well, short term, we have another major effort in the lab that tries to extend this selective approach to neurostimulation to not just to afferent or efferent, but to more specific fiber types based on their size or the diameter. I would say it's almost came to fruition recently, and I think we're getting close to submitting a manuscript on that. Another short-term goal was to establish and understand the long-term performance of these chronic implants. Now we know what to expect. We obviously have people that now can do it consistently in several animals. So in the intermediate term, I would say we want to test these therapies, vagus nerve stimulation-based therapies in preclinical models of disease, of which we have a lot here at Feinstein, especially in mice. And I would say a little longer term, and we want to translate that into therapies that can be tested in large animals like pigs and eventually in humans. The pig is a very good model for electronic devices that are human scale. Their vagus is very similar to the vagus of humans. Recently, one of Kip Ludwig's papers very well showed. The pigs also share a lot of the anatomy and physiology, especially on the cardiovascular side, with humans. So they are a very good model for testing bioelectronic therapies to treat cardiovascular diseases. And that's something we've been working on for the past couple of years. And we're hoping to get to a good point in the next year or two with that. And then, of course, the really long term is to start some clinical trials that use some or many of these principles of neurostimulation in human disease. And being part of Northwell, we do have access to a lot of clinical populations, to excellent physicians who have research interest, and they're always excited to try new things that obviously have merit. And that's what we're working on. And hopefully in the next 5 to 10 years, not just us, obviously, but a lot of groups around the world, hopefully will be translating all of these basic science discoveries into actual therapies.
Yeah, that's really exciting stuff. Hopefully that'll happen. Yeah, a few years, I guess, huh?
I mean, that's the cycle of innovation when it comes to medical devices. I would say 5 to 10 years, and that's assuming that the current technologies and clinical trials perform reasonably well. We learn on multiple fronts. We learn from the clinical trials what works, what doesn't work. We also learn a lot from preclinical studies. And we try to make better devices through this process. And that, takes years. The NIH obviously is involved, DARPA is involved on the more of the discovery side. But then once you get past that, you need commercial interest. I would say 5 to 10 years is a reasonable horizon for some of these ideas to mature to clinical therapies.
Yeah, hopefully. Well, Stavros, this has been very interesting. Oh, actually, you know what? I wanted to talk about, so people might hear an accent, and it's because you're from Greece. And recently on Twitter, you posted some stuff about the H-1B visas that might be canceled coming up and until the end of the year or something like that, right? Is that the case?
Well, it doesn't affect me because I'm a citizen, but it does affect a lot of other people that I work with every day, people in the lab that are here on H-1B visas. I mean, I understand COVID-19 is a very special situation, right? Like nobody really knows how to deal with it well. And I think that's true for the US as well as for other countries. But I'm not sure. I don't want to get into politics very much, but I think that speaking as a scientist, I think one of the many reasons that the US has traditionally attracted some of the best talent in the world in science was that at least students and postdocs and researchers didn't have to worry too much about their immigration status. If they found a position in an American university or research institution, it was relatively straightforward for all of this to happen, right? To get legal status, to not to worry too much about their visa and their entry, re-entry, all of that. I think making that a lot harder, as it seems to be happening now, might be a deterrent to some people from deciding to move to the States. A person from my home country, Greece, who applied to start a PhD here at Feinstein, and he did get accepted, but now he cannot fly because there is all this travel restrictions. So he was supposed to start in July. He's still in Greece. I think he might be enjoying the beach, so that might be good for him. But overall, I mean, it certainly doesn't help American science to have all these restrictions. But on the other hand, I understand that it's a very special situation. People are still trying to figure out what they should do, especially the politicians, which is a first because I think they always know what to do, or at least they think. I would say hopefully things will become a little more normal in the near future. I think the American science has a lot to gain from letting people get in without many restrictions. People who should be here doing science here, they should be allowed to.
Yeah, definitely. I mean, this is something that's also dear to my heart, like as a son of immigrants and, I've been an immigrant, I've been deported. And so these are all things that are very important. And I think the talent pool is being severely restricted, if you only look inside the US. It's hard to say what's the worst outcome out of the COVID situation, but I think this is one of the worst ones, actually.
I agree, because it will, you never know, but it might have long-lasting effects on how willing people are to leave their countries and move to a foreign country like the US without having at least the security that they won't be deported, like you said, or they won't be allowed to enter the country or be without a job for several months or without insurance and stuff like that. It can be good for the future of American science.
Yeah, definitely. Well, on that note, thank you so much for coming on. This has been a very interesting conversation.
Thank you very much, Ladan. I'll talk to you soon.
Hope you enjoyed the show and were able to learn something new, bringing together different fields in novel ways. Until next time on the Neural Implant Podcast.